This video covers false prediction number 5, protein evolution. Let's have a look. So the prediction is that proteins evolve. Proteins are evolvable. According to evolution, all life evolved, so proteins must be evolvable. They must have evolved. It seems to be a simple prediction. All of life's protein coding genes must have evolved. Or another way of putting it, evolution must be capable of creating all the protein coding genes. But we'll do well to clear away some confusions here. What is this not? Synthesized and manufactured proteins in a laboratory, those are not evidence for protein evolution. Proteins that may be generated in a modern cell are not evidence of protein evolution. Modern cells are capable of all kinds of adaptation, where, by the way, pre-existing proteins are required. Inferred protein evolution from phylogenetics is not protein evolution. Just because a protein coding gene is present in a genome of a species but absent in a similar species is not evidence for protein evolution. That presupposes evolution in the first place. Okay, so the falsification of this prediction. First of all, proteins are huge. And by huge, I don't mean physically huge. Of course, they're microscopic. But there are many amino acids in a protein, dozens, often hundreds, sometimes thousands of amino acids in a protein. And you can multiply by 3 to get the number of nucleotides in the protein coding gene. So proteins are big. Proteins are huge in terms of the number of mutations required to evolve them. Another problem is that there are a huge number of proteins. Not too many years ago, the number of protein families was estimated to be 1,500. A few years later, that estimate was up to 23,000. A few years later, it got up to 50,000 or so. In 2023, we broke through 100,000. There are a lot of protein families. There's a huge number of protein families, and probably more coming, as this paper states. We anticipate that this diversity in unexplored microbial protein space will continue to increase over the next several years as more novel environmental samples are sequenced. More are coming. Again, we're saying diversity has doubled, but in reality, it could be 3 or 4 or 5 or 10-fold more out there. So proteins are large and they are many. Evolving them must be really easy. As we'll see in a minute, those are going to be big problems for this prediction of protein evolution. Another problem is that proteins are orphaned. In other words, proteins are taxonomically isolated. This paper states that we noticed a strong narrow taxonomic distribution with more than 2/3 of the protein families being restricted to a single species or genus. So most of these protein families are found in a single species or a few species in a genus. Most proteins do not fall into the evolutionary common descent pattern. Continuing, these results reveal that a significant fraction of functional diversity remains taxonomically orphaned despite improvements in sequencing. Now notice that despite improvements in sequencing. What does that mean? Why do they say that? What's so special about sequencing? According to evolution, biological designs, such as proteins, should fall into a common descent sort of pattern. Orphans don't do that. So when orphans were first discovered, evolutionists thought, well, we haven't looked in enough genomes yet. These are not legitimate, genuine orphans. There's no such thing as a genuine orphan. They fall into a common descent pattern. We'll find them in other species, other sister, cousin species. They fill out part of the tree. We just haven't found them yet. So that's why sequencing was important, sequencing of new genomes. So look back at this quote here, despite improvements in sequencing. In other words, yes, we now have sequenced a large number of genomes. We've looked in a large number of genomes and we haven't found them. In fact, it's the exact opposite. Orphans did not go away, they increased. The next problem is that proteins are an all or nothing affair. In other words, you don't get a gradual build-up to a protein. Proteins are not made up of smaller components which themselves have a function. That's the way evolutionists need to describe things. The old phrase is, what good is half a wing? You have a wing on a bird, you have to evolve that wing somehow. So half a wing must somehow have a purpose. And evolutionists said, well, it's good for warming and so forth. So you could build up to get a wing. You have to build up to get things in evolutionary theory. The structures don't just appear in a whole cloth. They need to be gradually built up. We don't see that with proteins. Here is a chapter in a technical volume. The chapter is about evolution of proteins. And the authors write, there is a discontinuity in fold complexity between fibers being simple polypeptides. There is a discontinuity in fold complexity between fibers and all other folded domains. Also, fibers are structural, not catalytic, whereas the primary role of proteins is catalysis. So a discontinuity both in the structure and function when you're talking about protein evolution. Skipping down, most random polypeptide chains do not fold at all. So proteins need to fold up into their shape, globular proteins at least. And so you need to be foldable. And they're pointing out just that property alone. Most random polypeptide chains don't fold. And so you have native protein coding genes that fold, but deviate very far from that sequence, and you lose the foldability. Here they write, although many point mutations appear neutral, combining several such mutations usually results in substantially impaired folding. Thus, even though naturally observed proteins can fold, most of their closely related variants cannot. You lose foldability, you lose function in general as you move away from that native sequence. And so this suggests a rugged protein fitness landscape where you have these spikes where you have the native sequences. Get very far away from those and you quickly go down to a non-fit, non-functional sequence. Continuing, at less than 1 in a billion, and possibly much less, it is much less than 1 in a billion, it is fair to say that to an exceedingly good first approximation, polypeptide chains do not fold. It is amazing that life would be built on such a difficult property. The question of how folded proteins evolved is therefore entirely non-trivial. Indeed, there is no gradual buildup for proteins. Okay, next problem. Protein coding genes must have evolved early in evolutionary history over a relatively short time window, say tens of millions of years. You don't have billions of years. You don't have 4.5 billion years to evolve these protein coding genes. For example, we've talked about how the DNA code appears in most all of the extant species, and so therefore, under evolutionary theory, it must have been present in the last universal common ancestor a long time ago, early in evolutionary history. So the point is, the DNA code must have been present very early in the history of evolution. Well, the DNA code requires a small army of proteins. So proteins were around at the very beginning. Proteins must have evolved very early over a relatively short time period. As this chapter notes, this abrupt origin of so many proteins is comparable to the abrupt origin of animal body plans, which must have appeared abruptly in the Cambrian explosion. Similar problem. Evolution has these problems of high complexity, high diversity appearing abruptly in evolutionary history. Another problem we've talked about before is the circularity of protein synthesis. So in other words, if you were lucky enough to have a protein coding gene evolve by random mutations, it actually wouldn't provide any fitness value because it wouldn't be expressed. Why? To express a gene requires the protein synthesis machinery in the cell, which requires a small army of proteins. You don't have that if you haven't evolved proteins yet. You don't have proteins. You're talking about how to evolve proteins. You don't have the protein synthesis machinery up and running. So if you do evolve a protein coding gene, it doesn't provide fitness. It has to be expressed somehow. In order to express it, you need a small army of proteins. So you say, well, let's get the protein synthesis machinery up and running first. That'll solve this problem, right? Well, no, because you require a small army of proteins to be present. You've just skipped over the problem. So it's circular. Another problem is the astronomical odds of evolving a protein coding gene from random mutations to begin with. Let's go back to this chapter. Even at a defined chain length of 100 residues, now remember 100 amino acids is a relatively small protein, they can be much longer than that, but even for just 100 amino acids, the number of possible polypeptide chains is 20 to the 100 because there's 20 amino acids. So 20 raised to the 100 is an astronomically large number. It vastly exceeds the number of particles in the known universe. This paper looked at the problem of actually evolving a protein coding gene from random mutations. Now, it wasn't a full protein, it was only part of a protein, so it was a simpler problem. And they came up with a value of 10 to the 70th. In other words, they would need 10 to the 70th tries, 10 to the 70th attempts to evolve a protein, a simple protein in this case. That's an enormous number. That is a problem. 10 to the 70th is not within the realm of feasibility. You can't have that many tries in evolutionary history. And they say as much here. Such a huge search is impractical. Right, it is impractical. And implies that evolution of the wild-type phage must have involved not only random substitutions, but also other mechanisms, such as homologous recombination. Well, they're throwing that out there as a bone because they are evolutionists. They are presupposing evolution is true, so it must have evolved somehow, so let's just throw out some mechanisms. Homologous recombination isn't going to do the job. That's not going to give you 10 to the 70th tries. That's not helping the problem. It's just another mechanism, another type of mechanism to search the space, but it's not going to come anywhere close to solving the problem at hand. This is not the only paper that's come up with numbers like this. There's several other papers that have come up with similar numbers on the order of, say, 10 to the 70th, 10 to the 80th for relatively simple protein problems. So summarizing, this study concluded that 10 to the 70th attempts would be required. Furthermore, the protein was only a part of a larger protein, which otherwise was intact, thus making for an easier search. This conservative estimate of 10 to the 70th attempts required to evolve a simple protein is astronomically larger than the number of attempts that are feasible. And explanations of how evolution could achieve a large number of searches or somehow obviate this requirement are unrealistic and require the pre-existence of proteins and so are circular. For example, one paper, and this paper right here is what we're talking about, how much of protein sequence space has been explored by life on Earth. This paper, this study, estimated that evolution could have made 10 to the 43 such attempts. So they came up with this number, 10 to the 43. So in other words, the paper said, well, wait a minute, let's take a look at this problem of search and over evolutionary history, how much of the protein space could be searched. And they were pretty aggressive about it, and they came up with this big number, 10 to the 43. But the study assumed the entire history of the Earth is available, rather than the limited time window that evolution actually would have had. Even more importantly, it assumed the pre-existence of a large population of bacteria. It assumed the Earth was completely covered with bacteria. And of course, bacteria contain proteins. Clearly, such bacteria would not exist before the first proteins evolved. Even with these unrealistic assumptions, the result was 10 to the 43. Now remember, we're looking at something like 10 to the 70th as our requirement, or more, actually much more, but 10 to the 70th very conservatively. So this is 27 orders of magnitude short of the requirement. This isn't 27 times. This is 27 orders of magnitude short of the requirement. And we get there by this ridiculous 10 to the 43 estimate with all kinds of unrealistic assumptions. Furthermore, here's a paper by some protein engineers who concluded that not all protein designs are evolvable. This is a review paper, and again, they're concluding that some protein designs are not evolvable. Now keep in mind, these are evolutionists, and they're admitting that not all protein designs can be found by evolutionary means. It's just another piece of evidence adding to this problem that the odds are astronomically against randomly evolving a protein coding gene. Okay, the next problem is, or another problem is, that proteins have multiple functions. So what we've been talking about so far is a protein coding gene that has some function, that provides a protein that has some function. Well, it's very common for proteins to have multiple functions. So this just makes the protein evolution problem that much more difficult. Now, another problem added on to that is that protein coding genes often overlap. So you have a protein coding gene somewhere in the genome. That space on the genome often contains part of another gene, which may be read in a different reading frame. So you have multiple protein coding genes. So if you're going to evolve a protein coding gene with random mutations, not only are you going to have to find multiple functions, you're also going to have to, at the same time, be evolving another gene in another reading frame. These are just huge problems adding on to the previous problems noted here. And finally, there's the problem of inheritance. It's a little bit similar to the protein synthesis problem we looked at earlier, where you don't have fitness value for a protein coding gene. If you were lucky enough to evolve a protein coding gene by random mutations, it wouldn't be expressed because you don't have the protein synthesis machinery in place. Well, here you don't have the inheritance machinery in place. That too requires the preexistence of proteins, not to mention a population and so forth. So you need to pass it on to the next generation. Back to this chapter here, they first note that the whole generation of random polypeptides is a problem in and of itself. And then they get onto the inheritance problem. Let me read this section here. It is, however, entirely unclear how this starting material would have come about. No abiotic processes are known that can produce chains of more than 5 to 10 amino acids, and these processes are very inefficient. There is also the fundamental question of how the sequence information contained in the evolving proteins could be passed on, since this is an absolute prerequisite for any evolution. Big problem here, skipping down. The problem of passing on the sequence information, however, remains unsolved. Indeed. So there you have 10 serious empirical scientific problems with this prediction that protein coding genes are evolvable. You have multiple problems, any one of which is a very serious challenge. Most of these just demolish the prediction that protein coding genes are evolvable. So what is the outcome? What's the denouement? How do evolutionists respond to this? So back to this chapter here, the origin of folded domains, however, remains substantially unknown. Indeed. Here's another paper. Although the origin of the first primordial genes may ultimately be traced back to some precursors in the so-called RNA world billions of years ago, their origins remain enigmatic. So yes, you can talk about deep history and things that happen that we cannot observe, but it's enigmatic. It's not something you can show with empirical science. I'll get back to this in a minute here. Another paper, the chances of evolution finding proteins from a random start are highly unlikely. Indeed. Now, this back to this response of the RNA world. So this is a common response is, well, RNA did it. You talk about protein evolution, well, RNA did it. What do they mean by that? This RNA world idea is that there once was a different biological world that was not DNA based, it was RNA based. A completely different biological architecture which is no longer here, no longer observable. We have no examples of it. There's no empirical evidence for it. We can't even create it in the lab, but it existed sometime, and that's where all the heavy lifting was done. Well, not only is this just raw speculation, but the RNA world hypothesis itself has been disproven over and over again. I can't count how many times over the recent decades that this RNA world hypothesis has run into major problems, only to resurrect itself later on in spite of these problems. So you'll hear this appealed to over and over again. It's just raw speculation in spite of the empirical evidence. I want to finish up with just a couple more responses that you'll see. One of them is that the DNA code is an enabler of protein evolution. This paper talks about that, and how under frame shifting you get a new protein. So you have a protein, then you have a frame shift. So instead of reading A, B, C, D, E, you start with B, C, D, E. So you're off by one, for example. That's one of the 6 frames that you could have. And that gives you a completely new amino acid sequence. So you make a jump in amino acid space. But the DNA code has this interesting property that when you do have a frame shift, it tends to conserve physicochemical properties. And the classic example is the hydrophobic profile, the oily amino acids. So interestingly enough, the hydrophobic profile across that gene, across the protein sequence of amino acids, is somewhat conserved, not perfectly, but you get somewhat of a conservation when you do a frame shift. So the idea is that, okay, you've got this protein and this hydrophobic pattern and other physical chemical properties are really kind of key. They're special, they've been evolved, and now you can make a jump in sequence space, go to a new protein, but retain these special properties. So that's kind of illustrated here in this graphic, on part B of the graphic. The paper writes here, frame shifting could be an effective evolutionary strategy for generating novel protein sequences. A later review paper picked up on this. They write, frame shift mutations could lead to major jumps within sequence space, leading to new tertiary structured proteins, while at the same time retaining the already optimized physicochemical properties and secondary structure. And skipping down, it writes, frame shifting may be a powerful evolutionary mechanism for creating new proteins. Let's home in on that. This is quite a claim here. A powerful evolutionary mechanism for creating new proteins. That's quite a claim. If this is true, this could solve the problem of protein evolution. Well, what are the problems with this? Well, first of all, this whole claim that the DNA code has this special property of preserving the physicochemical properties is overblown. It's overestimated in this paper, this Bartonic paper. So here's research that I've done, looked at this, and if you take protein coding genes and subject them to a frame shift, yes, you do see a somewhat preservation of, let's just say, the hydrophobic pattern. That's what this chart is looking at, the hydrophobic profile along the gene, along the amino acid, the resulting protein amino acid sequence. However, only about half of that effect is accounted for by the standard genetic code, SGC. In other words, is the genetic code the cause of this? Well, no, it's only the cause of about half of it, this preservation. So the conservation or preservation of the hydrophobic pattern is not perfect. You get a little bit, and then only part of that is due to the genetic code. The rest of it is due to the sequence itself. So here I'm breaking the sequence into 3 different components. The amino acid composition of the overall sequence. You just take the overall sequence and look at the composition of amino acids. Then the sequence itself, which amino acids trail which amino acids, which are flanking, what are the flanking amino acids, and then the codons that are used for those amino acids. All 3 of those have their own contribution. So yes, there is a little bit of a special property to the genetic code, but there's also a special property to the sequences themselves. They contribute about half to this physicochemical conservation that you see. So the whole story starts to break down here. It's not just the genetic code. Furthermore, the hydrophobic profile is not itself sufficient. Yes, you do see correlations within a protein family, within similar protein structures. You do see similar hydrophobic profiles, but even then, there's quite a bit of variance. It's not, these physical chemical properties are not the magic to ensure that you're going to have a functioning protein. Another problem is that these frame shifts we're talking about are going to cause new stop codons to appear in the gene, and they also don't preserve the initial stop codon at the end of the gene. So let's look at what this means here. Back to the DNA code, you have 3 stop codons in the upper right: uracil, adenine, adenine, UAA, UAG, and UGA, the 3 stop codons. let's look at UAA and let's apply a +1 frame shift. So under a +1 frame shift, UAA turns into AA something. So you have AA something. What is AA something? Well, that's going to take you down 2 boxes to AAU, AAC, AAA, AAG. What do you get? You're going to get either asparagine or lysine. So under a +1 frame shift, your stop codon just turned into an amino acid, either an asparagine or a lysine. So that's going to screw things up. You no longer have your stop. So let's go back to this graphic back in this paper. Remember here under part B, I said I'd come back to this. You start off with the initial sequence, so it's in green there. You begin with an AUG, that's the methionine amino acid. It also serves as the start signal. And then you have the genetic information, and then it terminates with the stop codon. Okay, so then they go to this frame shift. They introduce a frame shift where the green turns to blue. So the blue part is frame shifted, and they show a couple of stop codons being thrown into the mix. You got a problem here. You got a couple of stop codons, which is something like that's going to happen. They also, what else do they show? They show the original stop codon is still there. That's false. You are not going to have the original stop codon still there. So that's a mistake. That's false. You lose that initial stop codon. Okay, that's one problem. But there's another problem. When you go to their next step is to say, okay, we're going to get rid of these stop codons. How you do that? Well, there's mutations are going to occur, and you're going to have these mutations, and these mutations are going to turn those stop codons into something else. And you could have a lot of possibilities there, but basically you're looking at other amino acids. You could also have another frame shift. And that other frame shift, that second stop codon, gives you another frame shift under some mutations, so you're back to green. Okay, well, first of all, so the problem is you have drift. So yes, you will eventually get these stop codons to go away. That will happen. But your initial mutations that come along are very unlikely to do that. So you're going to start building up, you're accumulating these mutations, and eventually you get lucky and you get a mutation that takes away the stop codon. In the meantime, you're introducing other stop codons. You're also just changing the sequence to garbage. It's just you're randomizing the sequence. And so you're not going to get back to something good. This is all just a just-so story that is extremely unlikely. They show the final stop codon coming back because they frame shifted back to green. But at this point, it doesn't matter. This is all irrelevant because you've got drift going on and more stop codons being introduced. So it doesn't just happen by magic. This isn't just a just add water world that this paper seems to suggest. Where is the review of this paper? Where's the peer review when we need it? Okay, so those are those problems. Finally, even if all of these problems somehow are circumvented, or they're not as serious as I'm suggesting, you still have this problem that you're requiring a protein as your starting point. This whole idea of enhancing protein evolution by the genetic code. The genetic code gives you this magical property that enables protein evolution. You require protein as a starting point. So again, it's not solving the problem. It's not genuinely solving the problem of protein evolution. And finally, one last problem is you can't evolve the genetic code like this anyway. The genetic code is, first of all, you can't really evolve the code. That's a problem in and of itself. We've talked about that before. But in this case, if you were somehow to be able to evolve the genetic code to get to this protein evolution capability, it wouldn't have any fitness value because fitness value requires that the benefit be within the lifetime of the organism. It has to be intra-lifetime. Protein evolution requires a much longer time period. So if you somehow evolved the genetic code to get this property, it wouldn't be selected for because it's going to take a long time over many generations. Your other option is to say, well, it didn't evolve. It wasn't evolved. This is just Crick's frozen accident. We just got lucky. Serendipity. That's evolution for you. So we're back to a theory that just is just drawing straws out of the air. It's just completely, this is just a just so story over and over again. So that's the DNA code as enabler. So one last response that you might see, hyperdimensional bridges. What's that? So this paper talks about these bridges that can happen because in protein sequence space or protein coding gene sequence space, it's a high dimensional space. You'll see these notional figures like shown here as like a three-dimensional space, but it's actually hyperdimensional. And the argument here in this paper is that you have interesting properties that emerge in these hyperdimensional space. And in particular, as suggested here, even though two proteins, A and B, might be separated by this valley, and a valley in fitness space, so your fitness goes down. If you go from A to B, you have to go to a lower fitness, and so it's not going to evolve. By evolution requires that you pass through increasing fitness regions, not decreasing fitness. So it's not going to work, right? On the left-hand side, it looks like it's not going to work, but the argument is there's a way around. And there's a large number of ways around. So you have all these proteins, and in this high-dimensional space, there's going to be a way around so that you can go along a path which is increasing in fitness to get from A to B. That's the idea in this paper. So protein evolution is solved. Voila. As they say in the abstract here, fitness maxima can be reached from almost any other phenotype without passing through fitness valleys. You can go from A to B usually. Okay, so and one problem with this is they're looking at structure only, and there's all kinds of other information layers in protein coding genes, and they admit that this is a problem. So they call this out. An additional sequence constraint is imposed on top of that required to specify the structure. Right. So they merely consider protein structure, but protein coding genes have multiple layers of information, not coding merely for structure. So that's a problem right off the bat. There was a Twitter discussion about this paper, which I joined in on and made several points. The evolutionists did not respond. So one of my points was, so if you, this back to that graphic going from A to B, you're going from one protein to another protein. Let's just take a couple of example proteins. Let's say you're going from triose phosphate isomerase to globin. What functional protein do you have at the halfway point? Because it's not triose phosphate isomerase and it isn't globin. So what do you got at the halfway point? Of course, that's a rhetorical question. It doesn't have to be halfway. As you depart the protein A, you're very quickly going to leave that protein, and you're going to have some other protein, and all along the way until you reach protein B. So there's a lot of intermediate proteins that you're passing through, all of which are functional, all of which have fitness. So there's a lot of proteins in this protein space, whether or not they are actually realized in evolutionary history. Presumably a lot of them are realized because you had to evolve all these proteins. But there's a lot of pathways. So that brings me to the next point here. There's the graphic there. Back to the graphic. So going from A to B, you have to go through a lot of proteins. My next point here, if the paper is correct, there must be many heretofore unknown functional transitional proteins on a pathway. So there's a lot of protein on one of these pathways. And if the paper is correct, there's many pathways for a given protein pair. It's not just one pathway. And there's many protein pairs. So in other words, there are many transitional proteins. They're never observed. There's no such thing. We've never seen any of these. So there's this whole world of unobserved, unempirical proteins out there that we know nothing about. So once again, back to a just so story, we have no evidence for this. And finally, also, we're back to this other problem where this idea requires the preexistence of proteins. Remember, in order to evolve protein B, you had to have what? Protein A. Proteins are required a priori. Proteins are required as a starting point. So you didn't really genuinely solve the problem of protein evolution here. So to summarize, we've seen a prediction, we've seen a very powerful falsification on many fronts. It really falls on its face, and we've seen a response that is entirely lacking. They're basically throwing up their hands and admitting we really don't know how this could have happened, and the ideas that are thrown out there are completely inadequate and do not answer the question. So we've got a very powerful falsification on this very clear prediction. What about the failed theory virtues we're looking at? In this case, I really don't see any of these theory virtues that can be saved. This clearly fails on all of these. The evidential accuracy, of course, fails. There is no causal adequacy. There's nothing here to explain this. Internal consistency, no. You have no gradual buildup in these proteins, no evidence of gradual buildup, but that's exactly what evolution needs, so there's an internal inconsistency. Coherences, no. You have theoretical components are tacked on to solve this isolated problem. Universal coherence, no, this is not consistent with statistical mechanics. Simplicity, no, we have a lot of additional complexity. Durability, no, it's another false prediction that does not survive. Fruitfulness, no, we do not have additional new discoveries by means of successful novel predictions and unifications. It's just failing on all of these. I won't spend too much time on this. It fails on evidential accuracy, causal adequacy, internal consistency, internal coherence, universal coherence, simplicity, durability, and fruitfulness. Religion drives science, and it matters.